Hello, welcome to Genetics in Pills. Today we are talking about the structure of DNA. So we started with Watson and Crick, and we remember Rosalind Franklin, co-discoverer of the DNA, death in 1958. So the usual picture of the DNA structure is that one you see in your monitor. Is sort of ribbon constituted by a double helix. The main backbone is composed by a deoxyribose, so the sugar, plus phosphate, so the alternance between sugar and phosphate, sugar and phosphate, sugar and phosphate. And this sort of ladder has these steps, and these are composed by the bases. A is paired to T, and G is paired to C. So this is the classical model of the DNA double helix. We have to say more about that. First of all, this is the chemical structure, schematized and simplified. So let's have a look at this side, okay? You see that the oxyribose is here, because at the carbon 2' you don't have the hydroxy but only a simple hydrogen. At the carbon 1' you have the bound with the base. It can be either adenine, cytosine, thymine, or guanine. Okay. Then you remember there is the 3' with the oxidryl, and the carbon in 5' bound with a phosphate group. So the nucleotide on the upper level is bound to the nucleotide of the level below by covalent binding between the phosphate and the oxidryl at the 3' at the carbon in the 3'. So if you can remember, in the previous pills we had this structure where is the OH here. This is the carbon in the 3', and down there could be phosphate, okay, like this. Here is the 5' carbon. Well, here this oxygen and this oxygen, they can be condensed to form a unique binding here, and so you have the covalent binding between the upper level with the level below. Okay, so it's this binding that makes robust enough the backbone of the DNA. So at a given point, this is a phosphodiesterase bound, is also called. Anyway, we have the bases that are protruding towards the other front, and the other helix is actually running upside down. So the 5' carbon is here, and the 3' is up here. So you can draw a sort of scheme where the double helix of DNA has two polarities. One is the hemhelix running from the 5' to the 3', and the other one is paired and is running in the direction 5' to the 3' in the opposite direction. And here there are the bases watching the one to the other. This other picture actually shows the 3D structure. It's not easy to detect the oxyribose and phosphate in this messy picture here, but you can see that the bases, pyrimidine or purines, are planar because they are all aromatic molecules. So they have a planar structure, and they are oriented with a plan at 90 degrees, so orthogonal to the axis of the double helix. And you can see they turn a bit every time. This is another picture, actually another scheme, even less easy to be interpreted. Anyway, we have the picture showing the groove, the minor groove, and here we have the major groove, the minor and major groove. One of the reasoning that helped to construct the good model was the distance between atoms obtained by crystallography and X-ray diffraction. If you pair two pyrimidines or two purines, you don't have the correct spacing between the two helix. They found that if you could pair two pyrimidines, there would be two room left according to the distance of the atoms in the molecule. If you instead pair two purines, actually there is not enough room for this operation. The perfect distance measured by crystallography was when you pair a purine with a pyrimidine. Thanks for watching Genetics in Pills.